Hey gang, welcome back to another exciting episode of Biology 1 Online. And today we're going to be talking about enzymes. I've got a special guest star with us today. That's right, Dobby the house elf. He's a great little helper. He reminds me a lot of enzymes because they're great little helpers too. So he's going to join us today for our presentation. So first thing we need to know about enzymes is that enzymes are proteins. They are made up of strands of amino acids that are folded up to be a specific shape. And they've got to be the right shape. Dobby? Dobby, what are you doing? Oh, come on, Dobby, don't do that. I mean, we got people trying to watch this video, man. You can't just... sorry, guys. I yelled at him earlier. He was kind of being a little butt, and I think he's just punished himself. We'll just ignore him. We'll just keep going. All right, so let's start off with this. So this is not an enzyme. This is hydrogen peroxide. And hydrogen peroxide is a normal byproduct of your metabolism, and it builds up inside your cells. And one of the problems with hydrogen peroxide is that it can donate its hydrogens very rapidly to things that surround it. And that makes it a pretty strong acid. And you probably think hydrogen peroxide, that can't be an acid. Well, the type you use at home is maybe 3 to 6% hydrogen peroxide. In the laboratory, we use 30% hydrogen peroxide. And this is what it looks like when you get some of it on your fingers. It actually breaks apart the cell membranes and kills and bleaches the cells. Bleaching means to strip all the color out of it. Have you ever known anybody that's ever colored their hair before, or bleached their hair? They didn't use actual bleach. They used hydrogen peroxide, and it stripped the color of the hair, thus bleaching the hair. So what does this do inside of our body? Well, again, it just causes a lot of problems for us, so we need to get rid of it. Well, luckily, there's this great enzyme called catalase. Catalase can take hydrogen peroxide and help it break down into water and oxygen. Now, it's important to note, hydrogen peroxide will on its own break down into water and oxygen, but it takes a really, really long time, like 50,000 years for it to happen. Right? It takes a long time. Sunlight helps the process go faster. That's why when you get bleach, it comes in a brown bottle. So that way sunlight doesn't help it break down. But catalase can speed this reaction up for us. It's an enzyme. You can see here it's made out of it's a tertiary structure of a protein. It's got a bunch of, quaternary structure of a protein. It's got a bunch of tertiary structures put together, which are in turn made of the alpha helix and the beta sheets and so far, which are all just chains of amino acids. But catalase can convert 90,000 hydrogen peroxides per second into water and oxygen. That's way faster, and thankfully we have that in us to help us break down all of that hydrogen peroxide before it does damage to it. So it just breaks it down into water and oxygen, and that's harmless to us. So yay enzymes. And Dobby's he's just a hot mess. Let's just keep going. Oh, now you're feeling better, getting with the groove some. Okay, let's keep going. Enzymes are very specific. So just like a key will only open specific locks, enzymes have a unique shape and only react with specific molecules. The key has to have the right shape. If it doesn't have the right shape, it's not going to unlock the specific door. And enzymes are exactly the same way. So we have a couple of parts we're going to label. Like we're going to label the substrate, the active site, and a process called induced fit. So the substrate is just what the enzyme will be working on. And the active site is where the enzyme and substrate will actually touch and fit together. Remember, that's going to be a very specific shape. Induced fit is when they actually bind together, they actually tighten up their grip on each other, so that way they get a nice strong action, so that way they can react together. So when we label our notes, we say, okay, well, here's our substrate, there's the active site on the enzyme, and that's where the two are going to bind together. So when they bind together, this is what it would look like. You can see that they're nicely snug fit together. So they've induced their fit, so they just pinch together. And this is kind of like halfway through the process. We're not done at this point. The enzyme has to actually do the work on the substrate. We'll see a little bit more of that in the next slide. I'm glad Dobby's in a good mood, though. We like for our house elves to be happy. So let's just kind of keep going. Yes, my beautiful little house elf. He's such a good guy. All right, so enzymes are catalysts. And what do we mean by catalyst? Well, most of us know that a catalyst essentially helps things happen. It's important to note that these catalytic enzymes, do two major things for us. First of all, they speed up a reaction. Second of all, they lower the cost of energy for that reaction. So they lower the activation energy. Think of it as like a coupon. You go to the store to buy something, and It costs 20 bucks and you got a 50% off coupon. Well, now it costs half as much. Enzymes do that for the reactions in our body. They take away the amount of energy we have to put into it in order to get it going, and they lower that for us, thus saving us energy. But it's very important to remember that enzymes are not used up during the action, and they can be used many, many times over. You don't have to have enzymes. The reactions in our body will occur without them, but it may take a very long time and a high cost of energy. So we still really need them pretty badly. But again, one enzyme can do its job multiple times, like that catalase did 90,000 per second. So it's not used up during the reaction. It's free to repeat it as many times as necessary. So let's continue on a little bit. Let's look at this site and Dobby, move! Move, I can't see. Thank you. It's about time you start listening. Oh, no, don't hit yourself. Oh, Dobby. Oh, I'm sorry, guys. I yelled at him. He doesn't like that. He's punished himself now. My bad. Well, look, we're just going to ignore him. Just ignore him. All right, let's keep going. So here we have a diagram of an enzyme and a substrate interacting. So here's the substrate, right? And it's going to bind to the active side of the enzyme, right? And so when it does, we call that complete piece the enzyme substrate complex. But as soon as it goes through its induced fit, and it gets all nice and snug on there, it's going to start taking the substrate and changing it into the products. When it does, we call that the enzyme product complex, and then the products will be released from the enzyme. So what have we done here? We've taken in some food into your stomach, and we've got some digestive enzymes that are breaking that food into smaller parts to so that they can be they can move into your cells and they can be used as energy or whatever we need them for. Dobby, man, I'm sorry, guys. Like, he does this at times. Okay, we're just gonna keep going. We're just gonna keep going. We're gonna ignore him. All right. So we've got to be able to turn enzymes on and turn them off. But first, before we do that, we have to make sure that they are actually in the right shape. And there are two major things that we need to do that. One is called cofactors, and those are the inorganic molecules. Things like iron, things that are not carbon-based. Remember we talked about organic molecules, we said it's carbon, hydrogen, and oxygen. These things don't have that. So this is iron. Heme, the hemoglobin found in your blood, it is iron-based. That is a cofactor that the proteins in your blood need in order to be able to transport oxygen. Other things we need are coenzymes. Coenzymes are just small organic molecules that we need to help the protein, the enzyme, get the right shape. Things like vitamins. Vitamins B, 12, and C, and A, and E, and E, and all the other fun letters, and K, and whatever you have for vitamins. They help it have the right shape. So why is that important? Well, later on when we look at photosynthesis, one of the things we'll talk about is these giant proteins that are embedded in the membrane of some of the organelles that help produce energy. So in here, it's just drawn as a big circle. But in real life, it looks a little bit closer to this. But this is not the proper shape for it. We've got some pieces hanging out. It needs to be a little bit tighter bound here at the bottom. It's missing its cofactors and coenzymes. So when we put them in, well, now we've got the right shaped protein. Now it can do its job, which is to spin around and help us produce energy. Here are some of the cofactors, and here are some of the coenzymes. They just help add to the shape and keep it going. So here are some examples of some minerals. Are these cofactors or are these coenzymes? If you take a second and look at it, are they organic? Are they based on carbon? Well, no, they're not. Potassium, iron, magnesium, phosphorus, these are all the names of other elements, right? So these are not coenzymes, these are cofactors. You don't have to know any of these, you don't have to memorize them or anything, but they're just there for you to kind of get a good example of what these things do and help it within our body. And then also we have some coenzymes. These are organic molecules that are going to help do things in our body and help proteins function and help enzymes do what they need to do. Again, you don't have to memorize these or anything. They're just here so that way you can see them and you can kind of get an idea of what's of what they do and what they help you with. All right, so good job on that so far. So we'll continue on with our types of inhibition for enzymes in the next video, and I'll see you then.